Dear students, today's topic is study on diseases of potato and tomato. Potato and tomato is most considered as vegetables crop in India, which is cultivated for its tuber and fleshy fruits respectively. A variety of pathogens like early blight, late blight, scab are attacked, which may greatly reduce yield. Now, let us discuss one after another the important diseases of the potato and tomato. So, first is the fungal diseases, and name of the disease is early blight of potato. Infected older leaves shows concentric dark brown spots at the center. Infected leaves turn yellow and sometimes dry out or fall off from the stem. In tuber, lesions are dry, dark and pressed into the tuber surface, with underlying flesh turning dry, leathery and brown. On stems, fruit tubers often show sunken and irregular shape and have the typical concentric rings and produces considerable sizes. Now, let us see about the scientific classification of this pathogen. Kingdom is fungi, phylum Deuteromycetes, class Dohc&Tm, subclass Pleosporomycetidae, order Pleosporales, family Pleosporaceae, genus Alternaria and species is Solani. Now, let us see about the character of this pathogen. The causal pathogen of early blight is the fungus Alternaria solani. The mycelium is haploid and septate, becoming darkly pigmented with age. Sporulation in culture can be stimulated by exposure to fluorescent light. The asexual conidia are born singly or in chains of 2 on distinct conidiophores. The beaked conidia normally possess 9 to 11 transverse septae. Based on the size of conidial chain or absence of the chain, Alternaria solani has non-catenati with long filiform beak. So, this is about the character of this pathogen. Next important disease is the late blight of potato. Infected leaves are identified by black or brown lesions and appear water soaked or have chlorotic borders. On tubers begin in crack, ice or lenticels, turns copper brown, reddish or purplish in color, produces a smelly rotten mass. Now, let us see about the classification of this pathogen. Domain is Eukaryota, phylum Heterocontophyta, class Omycota, order Peronosporales, family Pythiaceae, genus Phytophthora and species is Infestans. Now, let us see about the character of this pathogen. Phytophthora infestans is a member of Oomycetes, a group of organisms sometimes referred to as water moles, which are related to brown algae. The mycelium is hyaline and sinusoidal and the nuclei are diploid. Phytophthora infestans produces sporangia on sporangiophores. The sporangiophores are indeterminate. This stalk-like structure aids in air dispersal of the sporangia. Zoospores emerge from the sporangia. Zoospores are biflagellate with one tinsel flagellum directed anteriorly and one whipless flagellum directed posteriorly. So, this is about the character of this pathogen. Next important disease is the black scurf or stem canker. The inoculums is soil and tuber borne, which damage sprout emergence, underground stems, tubers and stolons of potato plants. The black scurf symptoms are common on tubers. The main phases of the disease are the stem canker, blight phase and the black scurf phase. Now, let us see about the scientific classification of this pathogen. Kingdom is fungi, phylum Basidiomycota, class Agaricomycetes, order Cantharellales, family Ceratobasidiaceae, genus Rhizoctonia and species is Solani. Now, let us see about the character of this pathogen. The causal pathogen is Rhizoctonia solani. The basidial stage is Thanatophorus cucumeris. The mycelium develops superficial scab-like and black sclerosia. Each sclerosium is accompanied by a superficial dark colored short-celled abundantly branched stout mycelium. No other spores are produced in asexual stage of the fungus. Basidial stage is mostly saprophytic. The basidia are formed on dead leaves or stem parts lying near the ground. The basidia produces thin-walled basidiospores. In turn, they can produce secondary basidiospores by repetition on germination. This is about the character of this pathogen. Next is the bacterial diseases. First one is the common scab of potato. This pathogen can be visualized when pathogens show small brownish spots which enlarge into water-soaked circular lesions forming large scabby areas. Now, coming to the scientific classification of this bacteria. Kingdom is bacteria, phylum Actinobacteria, class Actinomycetes, order Actinomycetales, family Streptomycetaceae, genus Streptomyces and species is Scabies. Coming to the character of the pathogen. Streptomyces scabies is a gram-positive, non-motile and rod-shaped bacteria. Next important bacterial disease is the bacterial wilt or brown rot. These pathogens show wilting, a streaky brown discoloration of the stem. On tubers, it shows a smutty appearance. Vascular ring and adjacent tissue shows browning coloration when cut. Now, let us see about the scientific classification of this bacteria. Domain is bacteria, phylum Proteobacteria, class Beta Proteobacteria, order Barcalderiales, family Ralstoniaceae, genus Ralstonia, and species is Solanacearum. And coming to the character of the pathogen, Ralstonia solanacearum is a gram-negative, rod-shaped, strictly aerobic bacterium. So, this is about the character of this bacteria. Now, coming to the viral diseases. First one is the potato leaf roll. Infected plant produces necrotic, turning brown and purplish and curl inwards towards the center of the leaf. Leaf rolling is more apparent and the entire leaf can become chlorotic and sometimes also has a purple discoloration. Now, let us see about the virus classification. It is under the group 4 positive single stranded RNA. Family is Luteviridae, genus Polarivirus. Now, coming to the character of this pathogen. Potato leaf roll virus is the causal pathogens and belongs to Lutevirus group. The variants are isometric, 24 nanometer in diameter and have 30% nucleic acid and 70% proteins. The genome is single stranded RNA, linear and unipartite. The most efficient and important vector of PLRV is Myzus persicki, which transmit the virus in persistent manner. The virus is also transmitted by grafting. So, this is all about the important potato diseases. Next is the diseases of tomato. First one is the Cercospora leaf spot. The characteristic symptoms include discrete circular lesions 3 to 5 millimeter in diameter with a necrotic center and reddish to dark brown margin, and in case of severe infection the petioles are also infected. Now, let us see about the scientific classification of this pathogen. Kingdom is fungi, phylum Ascomycota, class Dohc&Tm, order Capnodiales, family Mycospherulaceae, genus Cercospora. Now, coming to the pathogen character. The disease is caused by Cercospora baticola. The mycelium of the fungus is septate, dark colored and intracellular. The conidia are borne on the tip of the conidiophores and they are hyaline, elongated, filiform, multiseptate, broadly rounded at the point of attachment to the conidiophore and tapering slightly towards the opposite end. Next disease is the leaf curl disease. This pathogen produces leaf curl and reduced internodes, giving the plant a stunted appearance. Now, let us see about the classification of this virus. It is grouped under 2 single stranded DNA, family Geminiviridae, genus Begomovirus, species is TYLCV. Now, let us see about the character of this pathogen. The causal virus is a Geminivirus and belongs to family Geminiviridae. The virus has large number of hosts in several plant families. The variants are geminate 20 into 30 nanometer in size. The particles are found in phloem, parenchyma and in nuclei. The genome of the virus is single-stranded DNA, circular and in two parts known as DNA A and DNA B. The virus is transmitted by whitefly, that is Bemisia tabaci, in a persistent manner. Next important disease is the powdery mildew. The formation of first white, later grey tan mildew areas on both sides of the leaf is the most characteristic symptoms of the disease. Now let us see about the scientific classification of this disease. Kingdom is fungi, phylum Ascomycota, class Luteomycetes, subclass Luteomycetidae, order Erysipelas, family Erysipecae, genus Erysipecae and species is Betae. Now let us see about the character of this pathogen. The disease is caused by Erysipecae betae. Mycelium is superficial and persistent. Conidiophores are unbranched and erect. Conidia arise singly, hyaline, ovoid and 30 to 50 into 15 to 20 micrometer in size. Cleistothecia are globose, dark brown or black with 4 to 8 SI per cleistothecium. So, this is all about the important diseases of potato and tomato, their symptoms and character of the pathogen. Now coming to the conclusion of this topic. There are several diseases observed on potato and tomato caused by fungi, bacteria and virus. The important diseases include early blight, late blight, bacterial wilt, leaf curl. The initial identification of the disease can be obtained by symptoms produced by the pathogen to their host, and later the confirmation is obtained by the presence of the pathogen in infected area. However, for proper management of the disease or before application of the control measures, symptoms associated with diseases need to be identified properly, followed by the identification of the pathogens responsible for disease. Thank you.